Recording in progress.
Welcome back, everyone, to our final colligative property of solutions that we're going to discuss, and that is how the osmotic pressure of a solution changes when solute is added. And again, what it means to be a colligative property. Things like freezing point, boiling point, vapor pressure, these are all things that change when you add solute to a pure solvent. And that change is something that relates directly to only the quantity of solute present. So we're assuming we're all talking about ideal solutions here, right? The net amount and type of intermolecular forces are things that we can ignore, like we know they're present because we've made solutions. But osmotic pressure is another example of a property of solution that depends on how much solute is present. So why is that? Let's look into the definition of osmosis. So here we're looking at osmosis. I've shown a little diagram. Let me describe to you what osmosis is, like let's define it, and then we can get into the meat of what you're seeing in this diagram in a moment. So by definition, osmosis is the spontaneous or natural flow of water. And if we're going to be, not just 220 chemists, but in general, like chemists, including O-chemists, we would say solvent. Oh, no, osmosis is really only about water anyway, so let's just call it water. You're really going to talk about this in biology anyway. So osmosis, spontaneous flow of water across a semi-permeable membrane such that the water travels from an area of low concentration to high concentration. So there's a couple of new words in here. Let's highlight them and give them names. So semi-permeable membrane. If we're talking about a literal like biological membrane, right? It's an actual physical membrane instead of just like a piece of filter paper, which is something you and I might encounter in like our Gen Chem classes. But a simpler way to think about like a semi-permeable membrane is some kind of substance that selectively filters through some molecules. So you can imagine like a cell wall, if that were not semi-permeable, if it were fully permeable, it would not have this like selectivity in terms of what it could allow into or out of the cell, which would be a problem, right? Because cells regulate by saying, hey, I need this thing in me or I need this thing out of me. And I've got a bunch of these like embedded proteins in my cell wall that facilitate that kind of movement. So it's a selective filter. You can almost think about like a semi-permeable membrane as being like the bouncer at a club door, right? Saying, yeah, you can go, you can't. You can go, you can't. Okay, so you got this semi-permeable membrane. It selectively filters through some molecules. And in the example or the case of osmosis, we're talking about the flow of water. So this is something that will allow water to flow through, but typically not any like larger molecules. Remember, water is pretty tiny when we think about, especially biological systems, it's just got 3 atoms, especially compared to even some like solute particles, like in this case, we've got sugar here. We can very obviously consider that maybe water is what's going to be passing through this little filter here. Okay, so we've got a spontaneous flow of water through this semi-permeable membrane. And we have to consider like, why is it that water travels through the membrane in the first place? So I've said here that The water travels from area of low concentration to high concentration. Especially in biological systems do not tend to be stable when there are large differences across some kind of boundary, right? Like we as biological beings like to exist in a state of stasis, right? We don't like there to be huge drastic changes to properties that we might have or possess. And the same is true for just like a solution in general. So we have to remember that when we're talking about osmosis, the water moves to dilute the more concentrated side of the membrane. So it is not like, I know it's really easy to think, oh, I've got more things in my more concentrated, so then this is where my water is going to flow out of, because we just kind of generally as humans tend to like trend more with, oh, this is the direction of the flow. But that's not the case here. So the water is what's flowing through this semi-permeable membrane. So here, this green bar is our semi-permeable membrane. And we can see before osmosis here on the left. On the left, we've got our dilute solution. On the right, we have our concentrated solution. And again, solutions do not like to have this like degree of difference between their concentrations, especially if there's a way for them to achieve like this homeostasis or this equivalence. So pre-osmosis, we have this like separation or segregation of solutions based on their concentration. And then osmosis happens, which means that our water is going to leave the side that had less solute particles and more water, so that it can dilute the side that had fewer particles of water or more solutes is probably the better way to say this. So osmosis is where we see the water naturally moving to the more concentrated, double bracket, concentrated side of the membrane. Just as we said here. Moves to dilute, the more concentrated side. That is what it means when we're talking about the process of osmosis. All right. So osmosis is something that just happens, right? Then how does this relate to like a solution property, a colligative property? We mentioned like osmotic pressure, like where's this pressure term coming in? Let's talk about it. So there's a difference between osmosis and osmotic pressure. In the sense that like osmosis is the phenomenon that happens, right? The water transfers across the membrane from an area that is more dilute to an area that's more concentrated. And osmotic pressure is the amount of pressure required, REQ, to exert on the solution, meaning the more concentrated side of the membrane, to prevent water from flowing naturally, aka prevent osmosis. So we've got kind of this diagram here of what's called a U tube, like it's literally a tube that is shaped like a U. Down here in the middle, we've got our semi-permeable membranes. So this is the substance that will selectively allow water to pass through, but not our solute particles. So here, if we were to look at the initial state, this is like where we would predict water will flow, specifically this way toward the more concentrated side. And it will do so until we have reached what's called an equilibrium state, meaning there's like an equal exchange of water molecules or solvent particles across the membrane, because we've now like achieved our desired dilution across that membrane. So we'll talk about this one a little bit later. But in this conversation we're having, we're talking about osmotic pressure, right? Osmotic pressure. So osmotic pressure is the amount of pressure that we would have to push down on our solute on, here indicated by this red arrow, in order to prevent the water molecules from flowing and then creating a higher column, right, in our U tube. So like if we do have this flow, then we see that if we were to compare this height to this height, this delta H is due to H2O flowing through that membrane. So difference in height. Now, osmotic pressure. This is the pressure that we apply such that we are saying, hey, water molecules, like, yeah, you may want to pass through this membrane and flow toward the more concentrated solution side. However, I'm going to be exerting a force across the area that is this little disc in the U tube here that is going to prevent you, according to like fluid dynamics pressure, from even doing so. We have to maintain an equal height. So the osmotic pressure is the amount of pressure we have to exert to prevent any height difference in our U tube. So due to the water molecules flowing through a semi-permeable membrane. We have a symbol for osmotic pressure. This is capital Pi. And you can see that the definition for osmotic pressure is given down here. So if you wanted to calculate the osmotic pressure of solution, meaning like how much pressure you would have to exert on that solution to prevent osmosis from happening, this would be equal to M, the molarity or molar concentration of solute, times R times T times I. And I write my expression for osmotic pressure this way instead of this way, even though they're the same thing, because I like to remember this equation as pi is equal to Marty, MRTI. So we have M, concentration in moles per liter. R is our ideal gas constant. We are talking about pressure here, so some of our gas terms are going to come into play. So that is, if we need the reminder, 0.08206 liter atmospheres per mole Kelvin. So do not forget that. Rewrite it so it's not in orange. This is 0.08206 liter atmospheres per mole Kelvin. And since we are using the ideal gas constant, that means that T, our temperature, must be in Kelvin. So remember, if you need to convert from Celsius to Kelvin, Kelvin is equal to 273.15 plus your degree C. And then I is our good dear friend, the Van Hoff factor. Oh, that is not what I wanted. I wanted a highlighter. There we go. So I Van Hoff factor. So again, it's a colligative property. We care about how much of our particles are getting in the way of our solvent. So we need to account for that using the Van Hoff factor. This is our definition of osmotic pressure. And let's pay attention to the units, because I think we just assume that since we are in a chem class, our units are always going to be in atmospheres. And the answer is like, yes, but also no. So let's see here. This is osmotic pressure. So pi MRTI, let's just check our units. So M, mole per liter, times R. So that is liter atmospheres per mole Kelvin, times temperature Kelvin, and I is dimensionless. It's either like a whole number, or if you get into your upper division PChem courses, perhaps not a whole number. Even then, we'll talk in like a week or so, maybe even tomorrow, about how I could not be a whole number, but I digress. So let's look at these units. We've got mole over mole, liter over liter, Kelvin over Kelvin. This does in fact leave us in atmospheres. Whew, yay. So we have our osmotic pressure. We'll calculate in this equation in atmospheres. So pi is the pressure expressed in terms of atmospheres. So if you're given a different pressure, you are going to need to convert it into atmospheres if you want to apply this equation. Okay, so that is the concept of osmosis, and now the concept of osmotic pressure, aka the amount of pressure or force needed to exert per unit area in order to prevent osmosis from happening. So in our next video, we will, oh, just kidding. So here in our next slide, let's look at why you guys care about osmosis and osmotic pressure. So for example, here on the left, I've got some samples of plant cells. And if you are a biologist, you will recognize like how firm or stiff a plant cell is by describing it in terms of its turgidity. And you will probably maybe have also defined something like, I believe it's called a turgid pressure or turgidity pressure. A little bit different than osmotic pressure, but in general, we have this idea that cells maintain how stiff their plant walls are, their cell walls are. So plants maintain how stiff their cell walls are by allowing for water to either move in or out or whatever. So we have a couple terms here that I think you might encounter. We've got here in case A shrunken cell. So this is where the plant has let water escape, it's dehydrated, right? So this would have to happen only if there was a more concentrated solution exterior to the plant. So like the amount of solute inside the plant cell had to have been less than the amount of solute present outside the plant cell. That's the only way that our water would flow out of the cell. In example B here, we have a normal plant cell, which means that we've got this constant equivalent exchange of water between the interior and exterior, meaning like the concentration of solutes inside the cell is approximately equal to that outside the cell. So the water has no preferential flow from a more concentrated area to another. So like the osmosis is like a continual exchange happening back and forth. And then we have in case C here, what's called a turgid cell. So this is a plant cell that has become really swollen due to an excess of water flowing into the cell. And this would happen if you were to take your cell and, or sorry, your plants and put it in like some pure water, right? You would have to have a less concentrated exterior solution compared to what's happening inside the cell itself. So you might have seen some people that recommend you store things like celery or carrots in a glass of cold filtered water in your fridge. And the reason that they suggest that is so that you can increase the turgidity of your cell. Because if you've got, for example, a little jar and it's full of pure water, and then you put your carrot inside that water, osmosis is going to naturally occur and allow water to flow through the cell wall of our plant cells in the carrot, which is going to keep that carrot nice and turgid or firm. So it'll retain its snap, its texture. We like crunchy vegetables. Yeah. So this is why you might see people describing this process, like how to keep your stuff more fresh. It's not necessarily that it's more fresh, it's just you are increasing the turgidity. Another example, still thinking about biology, is again like actual animal cell regulation. So let's look at some new terms here. We've got hypotonic in A, isotonic in B, and hypertonic in C. These are very similar to what we've seen in our plant cell turgidity. In scenario A, hypotonic, this is where we have a solution surrounding our cell that has very little solute, right? So this would be like if we had our cell, which was more concentrated on the inside than it was on the outside, right? It's in an environment where water would spontaneously flow into that cell. So looking at it using an actual like little textbook diagram, you would have water flow in. Isotonic is again this equal and opposite exchange of water into and out of the cell. So we have like an equal concentration of solute exterior to the cell as we do interior inside the cell. And then case C, hypertonic, this is now where we've got a cell that exists in an environment where instead of being surrounded by something like pure water or a dilute solution, it is surrounded by a solution that has a high solute content. So like if you were to put this in salt water or in sugar water or in like an excess of protein, we would see that the majority of the water would then, via osmosis, leave the cell. And in both cases, A and B, so hyper above, right? Hypertonic, excess solute. Exterior to the cell, hypotonic, less solute. Exterior to the cell. But you can see, like I was saying, in case A and B, both are bad. Both are bad for our blood cells in particular, which is what I believe we're looking at here, a red blood cell. So we want to, our bodies are constantly in a process of trying to maintain like an isotonic environment so that our blood cells do not have an increase in water due to osmosis or a decrease in interior water due to osmosis going the other direction. I believe this is somehow related to blood pressure in general, but someone please correct me on that. I am not a person in medicine at all, so don't necessarily take me at my word. Okay, why you should care about osmosis. Let's talk about the colligative property that is osmotic pressure raising. So we've seen the equation, right? We know that osmotic pressure is equal to mRTI. This is how we calculate osmotic pressure. And the colligative property of osmotic pressure raising simply says that as the concentration of solute increases, so too does the amount of pressure needed to prevent osmosis or prevent water from flowing, because you can imagine if we had, and forgive my 3D sketch, it's not great. So here's our two beakers that are separated by semi-permeable membrane. If I have two solutions on either side of the membrane, and I want to exert some pressure down on these solutions to prevent osmosis from happening. In the example here on the left, if I had a really, well, let's see here. If I had fewer solute particles, then the desire of water to spontaneously flow across the membrane toward that solution is going to be a bit lesser, right? It doesn't have as much solution to dilute. So less spontaneous dilution. Now, if we compare that to the solution here on the right, where perhaps we have an incredibly high concentration of solutes, the natural tendency of water to flow toward that hyperconcentrated side of solution across this membrane is really high, meaning we have to exert more pressure down on the solution to fight the water that's trying to make its way across the membrane. So here, since we have a higher concentration of solute, we need more pressure to combat osmosis because more water is needed to dilute that solution on the left side of the membrane here compared to the solution shown here. Now, why is this a property you care about? So like we talked about why osmosis is important, but why is like osmotic pressure something that's interesting? So if you think about how human beings have stored or cured or preserved food for millennia, a lot of it has involved how we dehydrate our food, right? So like we create things like jerkies, we create things like dried fruits, dried vegetables, we dehydrate or powder things, and we also add like a bunch of salt, so we pickle them. Or in this case that I'm showing you down here, we add a bunch of sugar. So let's look at this as an example of osmotic pressure at play. So if I've now made some fruit preserves, this is a solution of fruit, sugar, and we have done our best to cook down as much of the water as possible without making this like a really disgusting solid, right? We want it to be kind of jammy and yummy. But it's a hyper concentrated solution. So if I had some natural bacteria, which we can not avoid having in our preservation process, we would find that the bacteria cells are unable to exert a high enough osmotic pressure to prevent water leaving the cell, which means that all the bacteria cells die. And thus the food is now no longer harmful, right? It had bacteria, but because we put it in a hyper concentrated environment, all the water inside that bacteria cell escaped. Now, if we had some cell that was like a really scary bacteria that could maintain its osmotic pressure, even in the presence of a highly concentrated solution, like our fruit preserves here, that would be a problem, right? If it had a really high osmotic pressure, it could prevent all of the water from leaving its interior and thus thrive and survive inside this hyper sugary concentration and hyper sugary environment, right? Which bacteria tends to love. So the bacterial cell wall acts as our semi-permeable membrane. And thankfully, osmosis is a thing that happens, and the osmotic pressure of these bacteria is not super high because this allows us to make jams and preserve our food. Okay. Now that we have a sense of like what osmosis is, what osmotic pressure is, and also now the colligative property, like the actual change in a solution that results from the effect of adding a solute known as osmotic pressure raising, we will calculate using our osmotic pressure example coming up in the next video. And then from there, we will move away from our discussion of physical properties of solution and start talking about more chemical reactions and how they actually how they actually behave in solution, like what's really going on in a chemical reaction. Because so far we think we know it's like reactants break the bonds, atoms rearrange, form products. And that's true, but there's more to the story. So that will be our next topic coming up when we discuss chemical equilibrium.